Yes, please. Chief Scientist and Engineer Mary O'Combe, would you like to say a few words? Mary. Mary, meet Linda. Linda, Mary.
Thank you, Adam. And I should say we're very lucky to have you all here, but particularly Adam. I was very relieved to see him on the plane this morning from Canberra. Last night was the Prime Minister's Science Prizes, and he was the most fantastic MC as he is here today. And I didn't think as it was ending last night that, he'd ever get away. So anyway, thank you, Adam, for doing this and for being part of the medical devices story. I too would like to acknowledge the traditional owners of the land, and I would like to particularly congratulate and thank our Minister. This is a really exciting and unusual fund. In my various roles, and over many years, I've seen a lot of venture capital funds in the medical area, in the non-medical area, and so on. I've never seen a fund quite like this, and I'm particularly honoured and excited to be associated with it. The Minister was very clear in her instructions to us as to what she wanted. She wanted the next Cochlear, the next ResMed, the next big company here. So the criteria are very carefully designed so that when we're looking at applications, we're looking to see if what we will fund will take or what the government will fund, we make the recommendation, will take an organisation on the way to being that we will be able to see the next Cochlear or ResMed possibly quite reasonably occurring. It's not straight venture capital. It's about funding great research, medical device research, but it's also about funding good companies that have a real future here in New South Wales, but companies that are going to be global, as are Cochlear and ResMed. It's wonderful and it's unusual. It's also a very broad definition of medical devices, and this is important. It goes through, so we're very well set up for the genomic revolution that's washing over us. And so devices taken in a broad sense. It might be hardware, it might be software, it might be special sensors. Most likely it's some mixture of the whole lot. We looked at last year's activities, we were very excited, and we were worried, as Adam said, would we see anything as good? And we definitely did. But we looked at our own processes, and one of the things we felt we needed was to have applicants much closer to the clinical base, the great clinical base of New South Wales, and it was a delight to welcome to our ranks Greg Keogh to have a clinical person on what is already a stunning committee. It's not only a stunning committee because the people are well qualified, they're wonderful people to work with, and I sit on a lot of boards and committees, and this is one I just really look forward to going to the meetings for. It's also, as I said, we're looking for devices that had all those criteria, but another thing the Minister has emphasised and we've kept in mind is that we don't only want to see it go global, we want to see it affecting health in New South Wales, so that these devices would affect all of us, help us with not only disease, but with disease prevention, and that also is in our minds. A lot of people to thank. I mean, we not only have ourselves as the committee, but we're very well served, of course, by Tony, the wonderful Tony Penner, Anne O'Neill, Ramona Sengupta and Rose Saputo, who from Tony's team in the Ministry of Health. They really do the very hard work. We come in and give opinions. The other thing is the subcommittee that Anne chairs that actually did the shortlisting for us. Anne O'Neill, Ben Smith, Stephen Thompson and Dr Jessica Wharton from my office. And they did a lot, there's a lot of work at that stage, and thank you for that. All those clinicians and experts who did the peer review for us and wrote quickly, and we often needed really top deadlines. Those who helped us shape, when we've decided which ones to go with, shape the actual form of the grants. And I'd particularly like to thank New South Wales Chief Pathologist, Professor Roger Wilson, who stepped in a very vital way that you'll be able to work out when you hear the things. Thank you, Roger. And to thank our financial advisors, O'Connell Advisory, Probity Advisors, the Procure Group, and the business advisors, Topaz Conway at Commercialisation Australia. Overall, it's a great team. On behalf of the committee, can I say thank you for allowing us to be part of what I think is going to be a stunning story. The Minister told us about last year's winners. We feel pretty confident we must have got something pretty right. We're going to hear about this year's winners, and Minister, it's fantastic that there's another round. I can see that we are going to see the next Cochlear, the next Red, maybe lots of them. Thank you all.
Thank you very much, Mary. Well, I think it might be time to meet the winners, the recipients of the grants this year. Minister, if you'd like to come forward, Tony, if you'd like to come forward too, why don't we present these very fashionable plaques, that I should point out are not legal currency themselves. You can't just take the plaque and say this is worth $2.4 million pay up, but they do recognise what you have achieved in receiving these grants. So first of all, let's call, and we'll show soon after that a video of the various winners, so that you'll just hear their names at the moment, and it'll soon be made clear exactly what it is they have done. But the first grant is going to Allegra Orthopaedics for their bone substitute material. Give Allegra a round of applause.
I think they should do it.
Yeah, come on up.
Yes, come on.
Smile for that camera straight down the middle there. And we've got Kiko Knee, the Kiko Knee Innovation Company, for their global scale project. The Sydney Children's Hospital Network is being funded for their paediatric bone rod. And Speed X for the in vitro diagnostic test for detection of viruses and bacteria causing meningitis.
Thank you.
Thank you, Minister. We'll meet each of those organisations in a couple of seconds, but first of all, we've got a brief video to play highlighting the projects that we are funding this year.
The Kiko Knee Innovation Company is dedicated to improving the quality of life of total joint replacement recipients. The company provides orthopaedic surgeons with dynamic, functional and patient-specific surgical planning and simulation solutions. The technology is currently focused for application on total hip and knee arthroplasty. The company's simulation platform uses engineering techniques derived from the aerospace and automotive industries. The Kiko Knee Innovation Company has developed a world-leading ability to produce accurate predictive assessments of in vivo implant performance during weight-bearing activity before surgery has actually occurred. This allows surgeons to customise the care regime for every patient with a focus on improving post-operative functional performance. A research team from the Sydney Children's Hospital Network at Westmead, under the direction of Professor David Little, has developed a revolutionary paediatric implant to treat children born with brittle bones or malformed legs. The new implant provides constant stability and support to the leg at the same time as correcting the underlying deformity. The novel design of their implant has stronger components and allows its rigidity to be substantially maintained as it increases in length. This extends the life of the device and minimises the need for replacement surgery, as is currently the case. They anticipate having the device available within 3 years. Allegra Orthopaedics is commercialising a synthetic bone graft medical device, which in one product presents highly sought-after physical, mechanical and biocompatible properties which are currently not being met in the orthopaedic reconstruction marketplace. The medical device is a bioceramic scaffold with outstanding potential for supporting bone regeneration in load-bearing applications. Preliminary studies indicate that it duplicates the mechanical strength, elasticity and bioactivity of bone grafts, but without the many recognised disadvantages of these current surgical practices. The Allegra Orthopaedics Bone Substitute, SRHT Garnite, has the potential to be the first commercialised synthetic bone substitute platform of products suitable for load-bearing applications. SpeedX is a Sydney-based company that has invented groundbreaking DNA detection technology, which it's using to develop tests to diagnose disease. The strength of SpeedX as a company is due to their MNazyme technology that is universally adaptable to detect any pathogen or cancer. MNazyme are pieces of DNA that can be tailored to detect different targets such as bacteria, viruses or cancers. MNazymes are easy to manufacture, and there are many companies worldwide that chemically synthesize DNA for research and for in vitro diagnostics, so they are readily available and are at low cost. SpeedX staff invented and developed MNazymes, and the technology is covered by patents owned by SpeedX.
There you go. There are our 4 grant recipients this year. So let's meet someone from Kiko Knee who'd like to come forward and tell us about the project. I'll get you to introduce yourself and what's your 25-word pitch on what your project actually does, and then we'll drill down a little bit. Who are you? What's Kiko Knee about?
Okay, my name's Beat O'Connor. I'm the Managing Director of Kiko. Effectively, 20 to 25% of patients who get a total knee replacement are functionally dissatisfied post-operatively a couple of years after the operation. So we think the solution is to actually assess the patient as an individual pre-operatively and modify their treatment and care regime.
That was about 27 words, but I'll let you go. So, okay, if I want a knee replacement, what do you do to me before I would have surgery?
Before surgery, we ask you quite a lot of questions, some psychological, some about your physiological needs. Are you a gardener or a golfer? Are you wanting to ride a bike? And then we take a CT scan of you, and then we build a patient-specific model. We throw that into a physics engine, and out the back of the black box comes an optimised knee replacement for you.
How different are people? Are most people's requirements pretty much within a couple of percent the same, or is there a massive range of different knees that you would need to produce over a population sample?
Every knee is very different, but the solution is not to make more and more implants. The solution, we think, is to actually customise the operative solution and then also the pre and post-operative care.
And yeah, because one of the important things is that post-operative functional performance that you were talking about. How quickly after someone's had an operation do they know if it's going to work or not? When do problems tend to materialise, and what would be the post-op care plan?
So the immediately post-operative issues are infection and reduced range of motion because of swelling. So that dissipates over the first few months. At 6 months, you start to see these first people who are starting to say, look, I know, doctor, you're saying my x-rays look fantastic, but this isn't working how I wanted it to work. So that 6 month to 2 year period is a critical intervention period. But realistically, all you're doing there is trying to fix something that's already gone wrong. So we want to take all of that change and put it in a cheaper environment, not in the surgical environment, but like a simulation environment, predictive analytic environment before the surgery, so that actually you don't even end up in that position at the end of 6 months.
And with some people for whom afterwards are not happy, is there a portion of people who are not happy even though things are actually working as well as they possibly could? They just didn't understand what the process was going to be like? And is that what you said also, because some of the discussion earlier is psychological as much as it is physical?
Yeah, there's a huge connection between the emotional state of the patient and their physiological side. So there's only a point to which we can take a catastrophising personality patient. We can only get them so far. Yeah. But everyone can laugh, but they do exist. Yeah. So you got to get that person to the best they can be. It's not about getting everyone to 100%. It's about saying what's the optimum result for you, and how do we get you there. Sometimes it's about surgery, but sometimes it's not about surgery at all. It's about all these other things.
And so with the money you're getting from the fund, how much are you getting and what are you doing with that? What's the timeline over the next few years?
Okay, so we have a commercialised product already. We have 20 to 30 patients a month are already getting our product. So our project's all about global scaling that around the world, and particularly going to Japan, UK and particularly the US. So we received a $2 million grant, and we'll be taking that straight away to the US, and it's now type thing. So we expect to be fully functional in the US in Q1, Q2 next year. But also, and I think it's really poignant, there's a lot of people in Australia getting knee replacements. There's probably about 16,000 in New South Wales this year alone. So if 16,000 people got our product, we need to hire a lot more people as well. So,
Very exciting times. Congratulations, mate. Well done, Bede,
you.
and the
Cheers.
team at Pico Knee. Who's going to come forward from the Sydney Children's Hospitals Network? Come on forward, David. Introduce yourself. We saw your face on the video anyway, but who are you and what's the 25-word pitch for the paediatric bone rod?
Hi, I'm Dave Little. I've worked at the Children's Hospital at Westmead for nearly 20 years, since it opened, and it's now part of the network. The paediatric bone rod is designed for kids, as we saw, that have congenital bone problems. But for those kids, we often need to fix their bones, but we need something that will stay and grow with them as they grow. Our rod has a number of unique features that allow it to be much more modular in how we put it in, simpler to put in, pretty much the same way you would put in an adult's rod who broke their femur or something, but it's also rotationally stable, which is the first growing rod to be rotationally stable, which was the trick that I was after.
Yeah, and that was part of the sales pitch you brought to the committee, that demonstration of that rod versus the current technology. So in simple terms, and we'll see them outside soon at the demonstration, how does the... Oh, you've got one. You just happen to have one with you. Fantastic. Show us how this works and doesn't work.
So you might have trouble seeing up the back, but this is one that would go into a thigh bone or a femur, which is why it has that little bend in there. And it just looks like a normal cylindrical rod, but as it slides apart, you can see that both in bending and in rotation, it's stable. And that's because of the ends, the profile of the rod. It's kind of a yin-yang arrangement where the rods fit inside each other. The traditional things are just car aerials where there's a male and a female component and they elongate like that. The male component in a very small child is always just bends. It's too small. These have been configured to have the maximum moment of inertia, which is maximum resistance to stress that you can get out of half a circle. So it's as strong as it can be for the size of the patient we put it in.
How did you come up with it? Because I don't mean this in a negative... It's one of those things you suddenly see it now and you go, that is so obvious. That looks almost simple in its brilliance. Where did the idea come from first, and is there still a lot of resistance for the traditional model? Come on in.
No, there was a clinical need because I'm not happy with the current system. We get complications all the time. It's a 40-50% revision rate within 2 or 3 years of those rods when they're supposed to stay for longer than that. So I wanted to try and solve that problem. The real problem was the male-female bit, so I thought we'll have to have... the whole thing just has to occupy the whole canal. I need the biggest component I can get into a small child, and then I've got to basically cut a circle in half. And then obviously you cut a circle in half and it falls apart, and then it did take me about 2 years to figure that you can cut a circle in half and it doesn't fall apart. And then that was it. Were
you at some vegan restaurant and you saw the little yin yang symbol? Because it's the basic message.
This is where Justin came in. Justin's my partner, who's got an IQ of unmeasurable. The yin yang thing was actually his contribution. I had other ways of fitting them together that wasn't as cool as this and wasn't as wouldn't have made each component as individually strong as they are. So we're a team, and it's a joint invention, and that's how you have to work.
You always need one hipster on your team, don't you? It's just across those sorts of... And with kids and brittle bone disease, I mean, any time you can avoid going back to surgery. I mean, surgery's not fun for anyone, but at any time in that sort of circumstance, you can make that better, both for the child and for the family. There must be...
That's
great
correct.
benefit
The other
in that.
thing is that this is the way this will be inserted will be minimally invasive. You can basically put this in through a hole this big.
Yeah.
And in our current innovations, we've worked out a way to make a hole down the middle of the rod, so we can even put it over what's called a guide wire and make it even less invasive. So the non-invasive component, the fact that it's rotationally stable, we won't need to put them in casts afterwards. Get them out of hospital faster, and the main thing that will make me say it really is a success is that we're not revising them, and that they only have to have, say, 2 goes in their lifetime. That would be the goal.
Have the people who have pioneered the car aerial technology seen yours and gone, yours is clearly better, we'll happily withdraw from the market and let you come on in and take our share?
Certainly all the surgeons I've spoken to who are confidentially in the know about it have said that they don't want to use that old technology anymore, and that this looks much more promising.
What is the grant? How much have you got, and where is it taking you over the next few years?
So we've got a $700,000 grant, which is what we applied for. About half of that is to do large animal experiments to show that the rod actually does grow as a lamb or a piglet grows, which is obviously necessary to prove before you start putting it into children. And the other half is business development and also prototype design. So this is a prototype. It's not what we would put into the patient. We still have to do some manufacturing improvements so that we can cut the thing out of a single block of metal. What we really don't want, any orthopaedic device that goes in with welds, on it, the weld ends up breaking. So we won't be putting it in patients until we've worked out a way of making it as each piece as a solid piece, which is also necessary. The other thing is getting the nails out. The other nails are impossible to get out. I've got a girl that came in yesterday, her nails are down here. We're going to have to cut both bones in half just to get them out. And so, again, this nail won't go in anyone until it's guaranteed that we'll have very simple, non-invasive ways of getting them out.
In 20 years you've been doing this, it must be very exciting.
Yeah, it's fun too.
It's great stuff. Well, congratulations. Give him a big round of applause. Well done, David. Really exciting stuff. Let's have come forward from Allegra now. And I've got two people coming up for Allegra because I want to examine both the science behind this, but also a quick insight into where the business is at. I think it'd be quite illuminating for you. Let's talk first of all about the device. Hala, explain to us who you are and what's the Allegra project.
So I'm Hala Zrakat, one of the biomedical engineering professors at the University of Sydney. We've developed something that is of clinical unmet need in the orthopedic field, and by inventing something that's purely synthetic, looks like bone and behave like bone. So we made the material to consist of ions that we know are important in bone formation. We also made it such that it's highly porous and interconnective, just like your spongy trabecular bone, but we can also make it at the edge to be the hard bone. We've put it in a large animal defect. As you know, if you have a defect in your skeleton, you're always applied load to it. So the most important part of this invention, and the critical one, is that we have a material that's highly porous and interconnective, but it's 100 times, and now with a 3D printing technology that we've also adapted now to make that material, it is so many magnitudes higher than that in any material available on the market. We've put it in a large defect, and we saw that it regenerated bone completely in its structure, bone marrow, cortical and trabecular bone.
Where were we before these 3D printers? This is amazing stuff, isn't it? So this is SRHT garnite.
Correct.
Is the stuff. Once you put that in me,
Yeah.
what happens?
We have not put that in humans yet, but...
What are you hoping will happen once you put some of that in?
That once we put that in, first of all, we don't have to use growth factors or cells, biologics. So that makes it much easier, much faster, cost-effective. But I'm hoping that once we put that in, orthopedic surgeons like David Little would not have to go back in and operate again. You'll get more bone forming, and the material eventually will resorb and become part of your body.
So my new bone will grow over
That
the garnite
material,
and
yeah.
form a single bone at the end of it.
Correct, and the material will eventually be resorbed and excreted off your body normally.
In the video we said preliminary findings. So where are you at in the path of this technology?
So thanks to the New South Wales Government Fund, this product now will be tested in large animals in shape, in large bone defects, and once results confirm, hopefully, what we found in the smaller animal model, the rabbit, then the company will take over.
Very, very exciting. Let's get the company in for a second. Tom, tell us about, because I think for a lot of the people, the innovators in the audience, the medical side is challenging and scary, but the company side is just as challenging in some ways. It's been a really interesting relationship between you guys over the last little while. Explain what's happening.
Yeah, you can see the passion that Hala has, and that's really brought Allegra Orthopaedics together with Sydney University. We've been working with them probably for about 6 or 7 years. We have an intern program, so a lot of the smart minds who are studying PhDs are coming through Hala's team into Allegra, working with our internal team and actually educating them and allowing them to move on to the next step. So now with the fund, it's fantastic news. We can now move to preclinical studies, and we've recently closed off capital raising, which is fantastic for us as well. So it allows us additional funds to really accelerate this and get into the marketplace as soon as possible.
It's a great partnership. What is the business plan over the next few years?
So the initial plan is doing the preclinical, as Hala mentioned. We're also working on transferring the technology out of a clinical lab into a manufacturing area. So Allegra at St Leonard's, we have 2,000 square metres of manufacturing space. We actually do manufacture medical devices. So we've got a team there working on our hardware orthopaedic products. So now developing a team to work on effectively the software orthopaedic products with the bone substitute material. Then we'll look at moving that from a preclinical into some regulatory approval in the US and also Australia to start accelerating the speed to market activities.
Very exciting stuff. Congratulations, Tom.
Thanks very much.
And Hala.
Thank you so much.
Very exciting stuff. Allegra. And up until now, you might have thought this was the New South Wales Medical Devices brackets bones only closed brackets funds. That's just the way it went this year, but we have a completely non-bone project in SpeedX. So please come forward. Give a round of applause, please, for SpeedX. In vitro diagnostic test for the detection of viruses and bacteria causing meningitis. Obviously, I know what that means, but if anyone in the audience doesn't. Who are you, Alison? Tell us about it and what's the pitch on the project?
I'm Alison Todd. I'm the Chief Scientific Officer from SpeedX. We've developed technology that can rapidly detect any gene associated with a bacteria or a virus or a human sequence that's associated with disease. So we can also work with oncologists to detect genes related to genetic abnormalities that affect the treatment of people. But our first test is for the viruses and bacteria that are associated with meningitis.
Now, this is, first of all, the M&A-zymes, aren't they,
Yes.
that you guys invented.
Yes.
What's an M&A-zyme?
An M&A-zyme is very short pieces of DNA that can be used to find the genetic target. When it does so, it sets off a little reaction that cleaves and generates a lot of fluorescent signals. So what you're looking for is far too small, but we can see a change in colour. So.
Because our current, when kids present and they may have any of a range of conditions that take too long to diagnose, the current practice is just whack 'em full of everything we've got in case it's the worst case scenario, yes?
That's correct. 90% of meningitis is caused by viruses, and they don't need antibiotics, obviously. The 10% that have bacterial form, those kids are really sick, or those adults are really sick. They need antibiotics, so we need to know. And if you're waiting for culture, you have to you just treat everybody, which is really not desirable. So.
Yeah, and that's what you're saying about this being targetable technology.
It's targetable to any gene of any disease state.
When people go outside and see the Lego demonstration, what are they looking at? What are you showing them there?
What I'm showing them is that we make these synthetic M&A-zymes. If indeed the gene matches those, then it lights up. So we have 3 genes: one a viral, one a bacterial, and one an irrelevant gene. If they snap onto the Lego model and it's a match, you light up, and that's exactly what's happening at a molecular level.
And so what money are you getting from the fund, and what does that help you do over the next little while?
We're getting 1.8 million from the fund, and what it means for us is that we can stop licensing out our technology so that everybody else on the planet makes money out of it, and we can start to develop tests in-house, and we can take it through validation and the regulatory product so that the money and the technology can help build SpeedX New South Wales and Australia.
Well, it's only fair because you invented it,
Yeah.
so you should share in the spoils.
If it gets out there, those are the spoils. Elisa McCartney is also here. It's a co-invention. M&A-zymes are a co-invention of Elisa's and mine.
Yeah. Well, congratulations. Give Elisa a round of applause too. Well done.
Thank you.
Great stuff, Alison. So that's SpeedX. So as I mentioned, when we wrap up here, there's going to be a little demonstration in the foyer of some of the technologies and the organisations. It's really been exciting stuff to be part of this year, as we've said a couple of times, but it is worth making the point that we came in this time around and then we're just blown away by the technology and also the really interesting business relationships that are forming around these industries and these devices. So on behalf of the fund, that brings us to the formal end of proceedings. Let's move outside. There's coffee and cake in the back corner of the room if you'd like, and then outside of the demonstrations to meet these great medical innovators and see their projects at work. So please, another round of applause for the Minister, the fund, our successful applicants, And also those of you who are here who are thinking about pitching next year and want to come and talk to people about the process and what went down this year and getting valuable insights, we're also available to be spoken to outside. So we'll see you outside in just a couple of minutes.